Okay, I'm assuming you have gotten an idea about what the project really focused on. You can pause for a minute. Let me quickly ask you one question. Anybody is open to answer this question. What specific biosafety issues do you see in the project? Anything, since this project is a very agricultural based project, do you see any biosafety issues? Anybody who has any opinions can speak up. Yes.
Since you're using a phage, I don't know how well you can control the evolution of the phage itself. So maybe the phage evolves to hunt more bacteria within the soil ecosystem or something, and that can cause a depletion of other bacteria in the soil ecosystem, which might cause some harm later.
That's a good point. Please also note that they are using a PDB, which is a delivery system, a bacteria that's specifically used to deliver the bacteriophage. And they're using this because they want to make sure that the bacteriophage is actually going and affecting the places that need to be affected so that the immune response is triggered, right? Um, we had a great point here. Anybody else who would like to add on to this? Yes. Parth, can you have one more mic? Okay, fine. Or is my voice audible without a mic? Is it audible? Okay, then I won't use a mic.
Yeah, I think in a similar vein to the previous answer, the fact that you have a dedicated delivery system for the phage could easily be misused. And there are also a lot of areas that could go wrong, which allows the phage to spread to other plants as well, which are not the initial sites of target.
The kill switch will be automatically activated once it I don't know, it goes on to a different I mean it goes back to the soil or attacks a different host cell apart from the plant. So that's how a kill switch works. I mean, you do have a kill switch, right? So
Uh, because you're adding a kill switch into the directly into the DNA. You're adding a DNA kill switch, right? The kill switch is directly added into the gene. What is the mutability ratio? How
Like how?
likely is it
Okay.
to mutate? Because
Right.
you're introducing with one, you're in. So I don't want.
Okay. Okay.
I'm introducing into direct large scale sector, and you don't have a proper delivery system. What I mean by delivery system is how you're going to be delivering it, right? Like how your pesticides being flown in from flights. One, we don't know the mutability of it and how it interacts with other bacterium.
Right.
Followed with that, we don't know how effectively mutations occur in the particular kill switch.
Okay.
Those are probably my concern areas.
Okay. So how it evolves and how it is being delivered.
No, specifically on this kill switch. Can the effectivity of the kill switch be altered by mutation?
Okay.
If so, what is the ratio of it happening? What are the chances of it happening? Why does it happen? That's all.
Okay. Perfect. So we have one valid concern here about how the kill switch evolves and mutates, right? And how fast does it happen? So it's a valid concern. And when you're looking at a project in such a way, you can again, now you can go and read up the safety part of their wiki. You can open it up right now and see what biosafety and what biosecurity policies that they had in place to ensure that this does not happen. Yes.
I was also wondering, since they were using a CRISPR-based system to repress the phage genome from expressing, what if the phage kind of, let's say, evolves around that CRISPR system, because that is also known, right? So maybe then the phage starts like lysing the bacteria before it even detects the given whatever they're trying to detect. So to begin with that, maybe that has some problem. Yeah.
This is again another valid concern. Anybody else who has any other similar concerns who would like to state their opinions? Okay, so we just saw this the bio safety part of the project, and I hope you can go through the implications or the measures that they had in place to control the same from happening. Right. Now, what do you think could be the broader biosecurity risks? So earlier on in the session, we differentiated what biosafety is and what biosecurity is. Do you think it can also have biosecurity concerns in the in the long run if not done properly? Right. Okay. So there is always a chance that it spreads and it keeps mutating in the long run. That's one good concern. Do you think it affects agricultural crops in any way, like in the long run? Changes to the, okay, especially the other plant that's being grown with it, right? That's one concern. So these are the biosecurity risks. Um, would you consider the project as dual concern, or something that has both a positive and a benefit? Are we on the same page with this? So it has both positives and negatives. Okay, I'll just, okay, I'll leave the mic here. I'll not use the mic. So when you're looking at dual use as such, when it comes to defining dual use for an entity that is not human, right? Say plant, becomes very, very difficult because again, it is a secondary entity. When you know that something affects humans, you have a closer understanding of how that can be very bad for human beings. But when it comes to plants, it becomes very difficult to define where that specific knowledge gap is lying. And as a result, you really have to think hard and see if a project does have a dual use research concern. Am I, by putting out certain research, for example, this one, or any other AIIM project documentation that you see on the internet, you also need to consider, is this too much information being put forward to maybe look at modifying systems that might cause some harm? Number two is this system good enough, or are the risks outweighing the benefits that the system might provide for people to work on, right? So some of these questions are important to know. Then again, I think the next question that you need to consider here is, would you implement the project in the real world? Why or why not? Is it something you would like to see happening in the real world? Of course, the answer is a little difficult because you can't just say yes or no. There is always, so if you look at the benefits, there are certain benefits. It's of course controlling the La Nina, and it's helping reduce the amount of crop damage that's being done.
Yeah, I think the process of having a delivery mechanism that protects the phage from external degradation mechanisms is something that can be generalized to a lot of things outside of this project as well. And I see a lot of potential negative use cases for it. So I think there should be a way to restrict information flow about that particular aspect of it, because it essentially protects the phage. And it could, I think that's one of the various aspects of the projects that could take like a negative spin. So I think in terms of information flow, you should restrict the amount of people who have access to details about how they did this delivery mechanism and such.
Can I ask? Yeah, I wanted to ask why exactly you've gone for a protection mechanism for a phage instead of fortifying the phage itself.
How do you fortify the phage?
For example, alternate methods for it along with the phage instead of adding another organism in totality, because adding another organism adds an element of variables and that could potentially lead to dual use. If you stick to one organism, it could be easier to regulate as well as probably is your project, I don't know, and probably use, don't know.
Okay, so one question here. Do you think something complex only has dual use implications? Don't you think a smaller entity can also cause something of a dual use concern?
To work on one, fundamentally choosing two organisms to work on one problem can potentially cause more damage than using one organism for one problem, is what I'm saying.
Okay. Do we have any other opinions? Any varying opinions? If you have any polarizing opinions, that would be nice. Anybody else? Yes. Okay.
Another safety measure that could be taken. The place where it's first implemented might be geographically isolated from the rest of the world, like an island or a isolated place, so that if there are some ecological impacts on that, it does not become global.
That's a valid take. Specifically used for labs that are engineering, um, who are making vaccines or like testing things with viruses, more prevalent in infectious diseases. So we've got a broad understanding of what biosafety means, what biosecurity means, and what dual use means. Essentially, dual use is when there is the potential for a certain biological entity, in this case, to cause potential harm if not handled properly, or if in the case that it falls into the hands of malicious actors, non-state actors, terrorists always can cause harm to the broader world, right? And there are certain organizations. Sometimes people can actively like weaponize it. For example, in the US, there was the anthrax attack in the early 2000s, where a lot of people were affected because of anthrax spores which were circulated. Um, and they did, I think 5 people died because of that. That's like the one of the primary or the earliest cases of said bioterrorism, or how a certain biological entity can be used for malicious intents. So the reason why we are having this workshop here is we really need to think about the potential dual use clauses and why we should really care, right? So for specific regulations of bioweapons or entities that can stem up from malicious use of biological entities, we have the Biological Weapons Convention, which is like this a treaty where they talk about why you need to regulate certain uses of biological matter and how you can do it. And I think there are about 185 countries that are signatories that are part of this agreement. However, it remains to be explored how this treaty can effectively be implemented across nations, because again, There are a lot of variations in how it is implemented, with of course countries like the US and UK and a lot of other European nations having stronger regulation and checks on dual use research as such, because they have a dedicated committee that looks into curbing certain problematic research at the beginning itself. And this is similar to how you send your projects or your research ideas to get reviewed by experts so that it can prevent harm from happening. Um, next slide. So next slide. Right. So this is the post survey. Um, we have about 25 minutes in excess, so you can attend this survey, and you can talk about what has changed. It's the same survey essentially, but you will have better clarity on what um, dual use is, what biosafety is, and what biosecurity is. So you can still you can give your opinions here. After this, we'll have some more discussions, some more general discussions about certain DURC aspects. Okay, I hope you're done with the form. Yes or no? Mostly done. Okay. You can save the form. You can complete this form because your responses are valid, and all responses are going towards the broader DURC survey that AIIM is doing to understand people's perceptions about how DURC impacts biological research and synthetic biology in specific. So once this is done, I'll give you 2 more minutes. We will have some discussions about DURC, how you look at the benefits and the risks of certain projects, and how there is a two-step verification mechanism, one before research and one after research, and how you can prevent excess data from being used for malicious intents. Okay, I think we can go ahead with our discussion. Hope most of you have submitted the survey. If yes, please give me a thumbs up. Okay. If no, please give me a thumbs down. Raise your hand and give me a thumbs down. Okay. How much more time do you need? Okay. Right. Are we done? Okay. Uh, okay. I'm assuming everybody has finished the survey. Let's now talk about the two-step verification mechanism that I was talking about earlier. So before you go ahead and undertake a research project, usually with a lot of dual use research concerns, you are required to submit a proposal and then get it verified and see if it has possible malicious intent applications, right? And a lot of these other countries, the US specifically does this. So the US and the UK, both of them have strong biosecurity policies in place, in that they constantly keep monitoring if there is something going haywire in the research process, and make sure that they are giving enough support for the research facilities to ensure it's not a biological entity is not being misused or deliberately being released. Um, and that is the first step in the research process, followed by inspections across the research journey till the end of the research. What happens after the research is done? It is likely going to be a research paper that will possibly look at the methodology and how it was done and be published in the real world. Often, since many journals are making it open access, it means that people are able to read this content and understand how certain things are being done, right? So in the likely event of certain information being sensitive, what people can do or what these journals can do is apply a check mechanism there and remove unwanted information at that specific place. So you don't have to talk in detail about what went behind engineering a certain organism. You can just say that an organism was engineered and remove excess clauses from the research paper itself that gives too much information, right? And that kind of prevents people from actively pursuing information that is on the web and replicating it. So if they are really genuine about about carrying that research out in their own laboratories, you can still reach out to the original authors and verify who these other people are who are reaching out to the authors, and then give them access to sensitive technology. Essentially, you're looking at how you encrypt data in a way that scientists or people outside the technical domains don't get access to information that can potentially cause harm. That's the two-step verification process, right? How do you then look at benefits and risks of a certain project? Ideally, if you sense that a certain project has a lot of risks, what you can do is discard that project altogether. Or if you think a project has minimal amounts of risks and a lot of benefits, you can look at mitigating the risks. That is the most efficient process that you can do. Maybe get again, citing this example, they had kill switches in place and a specific delivery system that could deliver the bacteria and then die once the process was done. That is one thing that you can do. Again, if you think that the okay, so we have gone across two criteria. One is when the risk is too high to be mitigated. One is when the risk can be mitigated. If you think there is significant scope for the benefits of the project to be increased, what you can do is you can look at strengthening how you can multiply those benefits. And I'm delivering this in a way that is understandable. I'm not using a lot of scientific jargon because it's a basic concept that can be used for emerging fields like artificial intelligence, where you can predict certain protein structures, where you can get multiple possible drugs or chemical entities for various amounts of for various things, for various diseases that you want to. So it is important that when you have access to certain technology like this, you learn how to use it properly with possible regulatory checks. Again, one final example. How many of you are familiar with how you order genes? I'm sure most of you have done this for AIIM, right? So you order genes, and then you specify the sequence that you want to. You send this order to the person who's synthesizing the gene, and it costs you about, how much does it cost? Maybe around 50,000, 60,000? Yes, based on the gene length, right? Anybody who has experience ordering certain genes? Okay, how many of you received the distribution kit from AIIM? Okay. What did you find in the kit? Okay. Anybody else who's seen the DNA, the distribution kit AIIM sent? I feel so bad giving the mic to you again and again. Nobody else is speaking up.
Yes.
Speak up. Yo, I just want to know what you saw in the distribution kit.
So we saw a lot of sponsor pamphlets, stickers, merch, merch.
On the, like, the sponsor pamphlet. You got
Yeah,
something
some
on the twist?
twist and even I think
How many
the
base pairs?
IDT.
Yeah, how many base pairs did you see written on the?
20,
Yeah, you
yeah.
had two, right? Okay. So what you do is you specify the number of base pairs. I'm not very well versed either, but I have some experience. So you specify the number of base pairs, you send it to them, they give it to you in about a month or so. That is in the good scenarios, right? When you don't have to wait for genes to do your lab work. So this is again an important emerging concept. So when you're handling genes and you're ordering base pairs, right? There is a lot of talk about having verification mechanisms in about who is ordering the specific gene. Is it a valid student, or is it an actual institute that is ordering a gene from your website? Or is it some third party non-state actor with possible malicious intent whose whereabouts can't be traced and could potentially lead to a bioterrorism event, right? So there, again, this is again an emerging topic when you look at gene synthesis, especially because it's becoming cheaper day by day to get these genes. And anybody who has access to an internet service and all these public gene databases can easily order one off the internet if they have the money and the wherewithal, right? So you will have to verify the person who is actually ordering this gene to prevent to prevent them from mutating or inducing certain other undesirable characteristics that might harm people in the long run. And that is again the importance of verifying a certain DNA synthesis and its implications for dual use. If you are doing that, you are reducing the risk for the DNA, the order to be misused. And this is crucial because if you are giving, and especially in this day and age when you have everything on the internet, right? Everything is open access. Everybody wants to access science and enjoy science that's being done everywhere across the globe. You don't want people to stop you from accessing the science, but I would rather say get people to be more cognizant of the the security policies that they have in place, and always be aware of all these policies so that you are not accidentally doing something that you should not be doing, right? I know I might be boring you all by saying, oh, you know, dual use is important. If you don't do it, you might be instrumental in, I don't know, creating another harmful bio weapon or reagent. It might sound a little dramatic, but there have been cases when nations have suffered because of negligence on the part of the personnel as well as the labs. So this formally concludes the session, but I would like to give a small anecdote of a certain uh, of a certain time when bioweapons were actually kind of used by nations. So this was the USSR, probably the 1970s, 80s. There was a certain pocket in the USSR very close to, yeah, in a certain pocket there was a huge anthrax breakout, and people, I think about 80 people died because of that. And people suspected, not people actually, the many nations suspected that it was deliberately released or accidental release from a lab that was actively trying to work on anthrax and look at possible bad mutations using that specific agent. Um, Turns out, USSR denied all these allegations for a very long time. Turns out, during the 1990s, one of the government officials accidentally revealed that, oh yes, we were working on a certain bioweapon, and that specific outbreak was accidental, or who knows, might be deliberate as well. So that is why we need to strengthen the policies concerning dual use in India, specifically since synthetic biology is still a field that is upcoming. You don't have a lot of strong DURC policies in place. So if you look at it, um, biotech, of course, has a lot of such reforms, such policies in place to ensure that it is being regulated properly. You have policies about GMOs and how they are being released into the environment. But for synthetic biology in specific, you have very, very less things happening, specifically on the policy front in India. This is crucial. If you want to learn more about syn bio policies and how DNA regulation, as I mentioned earlier, works, you can go ahead and look at what the US is doing. Certain countries in Europe are far ahead when it comes to regulating the research and making it safe for everybody. And those aspects can give you a little bit of clue about how or where India can improve on the synthetic biology front. Which is why we are here to deliver this session, because we figured that in India, AIIM is one way synthetic biology is growing, and we felt that teams needed to know about the DURC problem, or how best to regulate it in your research. And as a result, we always recommend teams to document the safety aspects that they have in lab, possible security measures that they have in place to reduce the risks in the lab, and make sure that you are conducting your research safely. I might have bored you down nicely for 1 hour. I would have loved to see more participation from your end, but I hope there was a lot of new things that you learned today. Maybe some of it can help you explore more about dual use, the Biological Weapons Convention, how it closely interlinks with governance, policy making, and geopolitics, if you're interested in that specific realm. You can, of course, go on the internet, read a lot of things about how biology as such has been used for previous acts of terrorism. I'm not going to mention all of that because it's going to be an information hazard. I don't want to be giving you bad ideas. But you can definitely go and read up on that, because it's a very fascinating aspect. You don't just have nukes going on around the world, but you have possible small biological entities that can be carried anywhere, possibly holding the potential to create a risk, right? And you don't want that happening. So when you conduct research, even though it is a small research for a small project, make sure that you are taking all necessary precautions, necessary regulatory mechanisms to prevent certain biological entities from being misused, right? With that, I'd like to conclude the session. Thank you so much, Hannah, for helping me out with this. Yes. 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 Okay, great point. As I mentioned earlier, if you see that the benefits are outweighing the harms, right? It can be valid to pursue that project, but whatever 0.99, 0.09 risk that you still have, how can I mitigate that risk, right? It is not about having that 0.09 chance of a risk, because even that can cause a lot of harm, technically speaking. So you don't want that happening. So you need to have proper measures in place for that to not happen, right? So in this case, you will look at minimizing the risk as much as possible while increasing the utilitarian benefits of a certain project, and see if it can help solve the problem that you're tackling, right? Any other questions, doubts? I'll quickly pass it on because we have professors waiting for the next talk. Well, with that said, if you have any other questions, you can reach out to both of us. We'll have Sasha joining us tomorrow. So we'll have 3 ambassadors here. And any doubts about iGym, anything else that you want to know, you can always reach out to us. Thank you so much for your patient listening. If you feel like interacting after the session, do come and talk to me, because I have certain things about DURC, because I really like reading up a little about this topic. So yeah, see you in the lunch break, hopefully, with more opinions, more polarizing opinions that I can then debate about. Thank you so much.
Thank you so much to both the ambassadors for the really great talk that they've given about dual use research. We would like to present you with a small token of our regard for helping us conduct the event, and of course for this. So our next talk will be by Professor Deepshika Chakravarti. She is an esteemed microbiologist who has over 20 years of experience working at the Department of Microbiology and Cell Biology at IISc. She'll be speaking about the topic of her talk is microbes everywhere, not a niche to spare. So I'll hand over the podium to Dr. Deepshika. And
and somehow this talking in front of an iGEM team has always been a pleasure. I did the same last year also. There are two reasons for that. The first is I'm very fond of the colleagues who work for iGEM. I was mentoring the iGEM team for past 10 years. I just stopped that because the new colleagues came and they started. Then I was also involved in mentoring other ICERs. ICER Trivandrum was, anybody from ICER Trivandrum? Not yet? Hi, you are there, hi. So the question here is that, working with the young minds and very novel ideas, and then vetting those ideas and getting to understand how things work, it has always been a pleasure. So I have learned a lot. Now today what I'm going to talk to you all about is my topic of choice, which is microbe. I am a microbiologist by training, so I did my MSc, BSc, MSc microbiology, followed by my PhD in infectious disease. Then I just took off. I was a Humboldt fellow and I took off to, I just do it by hand. Na Parth, is there any, something is there? No, not this. Is there a pointer, or is there a pointer? No, don't worry, I'll just do that. As you know that AIIM, nowadays I am seeing a lot of AIIM projects which are quite different. They don't involve a lot microbes, and there are many of these projects, they do not deal with making these bio bricks. Of course, bio brick is one of the most important part here. But whether you work in the field of physics, chemistry, mathematics, these three fields make actually biology what it is, and so is with microbes. Our microbiology knowledge is very incomplete without the involvement of these three subjects. So I always tell my young colleagues that if you have left physics, chemistry, and mathematics, somehow, you know, I'll not be very interested to make you pursue in microbiology, because really microbiology is something which needs a very deep knowledge of all these three subjects, and then you can make it very interesting. So what the title here is microbes everywhere, not a niche to spare, which is true. Now this room is filled with microbe, you can sample it. We are exteriorly covered with microbe, our interior is with microbe. And if you really look into the origin of life, or the earth, which came 4.3 billion years ago, just 1 billion year later, it was microbe. So microbe is the one which make the very obnoxious earth livable. So cyanobacteria is the one which indeed made this earth livable. So we start with microbe. So from sponges to the human being, each one of them, they have their flora. Even the primitive sponges, they have come with their flora. Now imagine if we do not have our gut flora, what is going to happen to us? So the gut flora consortium is so important. You are what you are only because of the microbe that is within you. If we are getting rid of that, then we are also gone from this universe. And so are the other beings like mosquito, honeybees, plants. Everybody comes with their microbes. And what we are doing at the moment, now if you look into this origin of life, everything began from something known as a primordial soup. That is the unknown common ancestor. This is very textbook picture, and from there the tree arose. But when the tree arose and microbes are everywhere, what we are doing very wrong now is compromising their right to live. Now the right to live is for every single human, every single species on earth. It's just not the human being, right? It's every single species, and so are the microbes. The way we are compromising the right to live to microbes is the reason for us to tackle a very serious pandemic of the so-called AMR, that is antimicrobial resistance, which is so much talked about. Who made this AMR? It's none other than the human beings. This concept of antimicrobial resistance. So how do we compromise their right to live? Today, what I will do is I will speak about the work that our lab is engaged in for almost 20 years now. We work with a so-called microbe of interest, and that is a pathogen. So that pathogen is a salmonella. I will tell you that why this pathogen is so interesting. Unlike E. coli, and you have heard about E. coli, Escherichia coli, all of you, E. coli and salmonella, they were very close relative. They just diverged couple of million years ago, and E. coli remained as a kind of a borderline between a good bacteria and a bad bacteria, commensal and a pathogen. But salmonella became a strict pathogen. And why they became a strict pathogen? Okay, they are gram-negative bacteria, so they are facultative anaerobe. That means within the system, within the gut, where the oxygen is very low, so they can grow in this microaerophilic condition. And they are very smart. You can put them in any kind of condition, any kind of insult, they are going to grow. They are rod-shaped bacteria, and all of you who have done your maybe first semester, second semester introduction to microbiology, you know what is the shape. At least my colleagues here know, right? You all enjoyed why there is a shape. Why do you think that the bacteria has a shape? What are the different shape of bacteria you can think of? Cocci, rod, you give it a curvature, and then many of these small curves. Why do you think that these bacteria, they have a shape? What makes them to have a shape? What makes the mammal, yes. Again, little bit. Surface area to volume ratio. No, surface area to volume ratio, that is the smartness of the bacteria to have their everything much faster.
Okay.
Yeah, so the surface area to volume ratio is very high, and that is why they are what they are in their ability to sense the environment and do very fast activity. So E. coli is dividing every 20 minutes, so fast. Cell cannot do that, right? But what makes the shape? Yeah. Yes. I agree. But then what gives it a shape? What is a molecular mechanism behind the shape? What is a what gives the shape to a cell? Say suppose if I say epithelial cells, what gives it a certain what is the huh? No, no, no. The shape I mean. Very good. Cytoskeletal elements. So you have a corollary cytoskeletal elements in bacteria. That is what gives it a shape. So the similar kind of cytoskeletal elements what you find in the mammalian system, you have a counterpart. It's not the same, but you have a counterpart. Say FTSZ, crescestine, which gives it a vibrioid shape, and then you have MREB, which gives a rod shape. Yeah. So this is why your bacteria is rod shape, because it has a protein which gives it a rod shape. If you mutate it, though it is very difficult to perturb the shape, but it's an interesting experiment to do and see if you conditionally change the shape, what will be its effect on the virulence, or if it is environmental bacteria, whether it is going to do the good thing or the beneficiary things that it is doing, or if you make the shape in between the two, or if you make it like a pleomorphic, like changing the shape, whether still it is going to do the job. So, okay, Salmonella is strongly a rod-shaped bacteria. It has a peritrichous flagella all over. Again, a very interesting point. Everything is a thought process. Now, if you have a bacteria with a peritrichous flagella, that is, it is all over. You have a bacteria sometime with a unipolar flagella. You have a bacteria where you have 2 or 3 flagella at one end, and you have also a bacteria with this. So, they are coming with different kind of flavors. Each flagella to be assembled, they are using their molecular secretion needle system. So, this is an amazing secretion system needle which the bacteria make, and those needles are assembled here through which your flagella is formed. So, this is a lot of physics which goes on to understand that how actually there is this hard cell wall is being perturbed to insert the needle structure. What is the force exerted? What are the kinds of motor proteins which are there which will give it a flagella? So, an amazing thinking, but at the time when the bacteria wants to get inside, it will not go with its flagella at all. It will shed off its flagella. So, you look at the diversity what is happening. So, these are peritrichous flagella, and they do not form spores. You know that the spore is formed not by the gram-negative bacteria. We still do not know the answer. It's only formed by the gram-positive bacteria, and these spores are long-living entities. Why did we take some 4 decades to disprove the theory of spontaneous generation? It's only because the spores were not being discovered at that time. The moment spores were discovered by Bajering, a soil microbiologist, and John Tyndall, they realized that yes, this entity, when it's given a chance, will get into the vegetative form and go, and that was the end of the era of theory of abiogenesis. Right. So, who discovered this part? Path, something wrong, got stuck. Should I do this? I should do this. So just right, it gets frozen. Okay. So the credit goes to Daniel Salmon, and that is why the name Salmonella. For Salmonella causes typhoid fever, and that has been put under the global priority pathogen list now, along with the escape pathogen. Salmonella also have a serovar which causes local gastroenteritis, and that numbers are huge, 2,700 serovars. So it is really a very, how would I say, very nasty pathogen. And interestingly, this particular paper, Nature, Ecology and Evolution, showed that the Aztec population way back in 1545 and 1576, the Salmonella paratyphi killed 7 to 18 million people. It completely wiped off this Mexico, highland Mexico. And that was not because of tuberculosis or plague. That was supposed to be, you know, the prime organism to create a renaissance in the disease biology. You know about the Great Fire of London, where how the plague was eradicated because there was a fire, and similarly the smallpox, chickenpox. But this one was done by Salmonella paratyphi way back then. So indeed it is a problem. If you look into India, we are the hub of the Salmonella infections, specifically the typhoid and the paratyphoid fever. So colleagues, there is a one word of advice to all of you. This particular bacteria infects you through the fecal-oral route. So contaminated food and water. Just make sure to wash your hands before you eat. Do not eat raw vegetables, because what we also proved that I said, na, microbe everywhere, not a place to spare. Even the tomato plants get infected systemically by this pathogen. And the big outbreak of the tomato contamination in California, it was not a post-harvest problem. It was really the Salmonella from soil got attracted by the root. There was a lateral root opening and the Salmonella just got infected. The tomato looked as red as ever, but when you cut it and put it onto the agar, SS agar plate, you see the growth of Salmonella. So the point here is that it is better to avoid raw vegetables. It's very sensitive to heat and you can kill the Salmonella by heating. Now, Mary Mallon's story, how many of you know about this typhoid Mary? Yeah, great. Mary Mallon was a only known healthy carrier, and she was the one who always got perturbed whenever the officials wanted to, take her to isolation, because she was a healthy carrier. But she was the woman who spread the typhoid through her cooking. And the favorite food that she cooked was peach, a fresh peach with ice cream. And she had spread the typhoid through under her nails, cooking through her nails. So in the end, she was isolated in Long Island, and after that, she never mixed with the people. And once she was isolated, the entire incidences of typhoid fever came down. So Mary Mallon became very famous lady, and she was also known as a Typhoid Mary. There was very interesting way by which they treated typhoid. Some 2000 years ago, Antonius Musa, he was a physician who treated the emperor by cold bath, because Salmonella has a very high, typhoid fever is a very high pyrexic charge. And so the first person who was credited was Thomas Willis way back in 1659. So yes, indeed it is the pathogen which we must take care of. Do not allow this pathogen to get inside. Slowly we are understanding that it is just not constricting or restricting itself to the small intestine. This disseminates to the brain. So there is something called as an extraintestinal infection leading to meningitis. Now our collaboration with NIMHANS is showing that many of the individuals who get infected with Salmonella typhi in their young age show the abnormalities in the brain. There are patterns, so there are signatures which are somehow retained. Mood swings, bout of depression, some signatures, they continue to remain so in spite of the bacteria not being there in the system. So my request to all of you is that always be careful in what you are putting in the mouth, wash your hands and take care. Now the Salmonella enters through the contaminated food and water, and if you look, this is the section of the small intestine. Again, very smart. The entire gut flora is lined with a very thick mucus. See, there is no way by which the pathogen can attack. Now if this is the small intestine, you will have 150 micrometer thick mucus. And on top of this mucus, you will have this flora. And this flora is somewhere around 230,000 species. It's like a mat. What on earth any pathogen that comes inside can cross and get it here? How? How they can get it? Now somehow these intestinal cells also have a cell called as a M cell, membranous cell. And these cells, not much worked out on M cell, but these cells, if somebody can characterize, this is an amazing discovery, because these M cells can be a portal of entry for many things. So Salmonella takes advantage of that, and this area is no mucus. So it's like simple. It's like, in the childhood you have, you did that slip thing. You sit on, what do you call that, slip, and then you slides. It's just like a slide. They come here, beautifully slide through. There is absolutely no inhibition. And then they get into the so-called the underlying layer. Now this is the underlying layer of your intestine, where you have macrophages, T cells, B cells, dendritic cells, and your bacteria is taken up by them. These cells would have killed the bacteria, but they don't. They are used as a carrier, like a like a vehicle to transfer now from the site of the intestine to the other organ. So instead of getting killed, they are using it as a vehicle. So the question is that, sorry Parth, it is getting stuck again. No, it came. I know what to do now. So 20 years of the research in the lab kept us really busy in first of all understanding that which all system can it infect. These cells, that's what I said, that there is, unlike viruses which have a permissive cell line, like if you talk about a COVID virus, the COVID virus will not infect any cells which does not have the ACH2 receptor. It needs that. Salmonella can infect everything and anything. Anything you put, this will get inside. Once it gets inside, it resides in something called as a salmonella containing vacuole. So this is like a studio apartment for salmonella. They like their apartment very much. This is a cell, and the moment it gets inside, the first layer is that of the plasma membrane, and they will live here. They just live here. They do not quit this so-called salmonella containing vacuole. And this vacuole had become extremely important for them to grow. When this vacuole, when the bacteria wants to divide, they don't do like this, you know, that multiple bacteria coming in a one vacuole. No. You will be surprised to see that when the bacteria divide, the vacuole also divides. So there is a division of the salmonella along with its vacuole. Now, salmonella division, or for that matter any bacterial division, you know, E. coli division, every 20 minutes they divide, you will know that. But why the vacuole divides, how the vacuole is dividing? So this entire story, which actually began from our lab, now had come to the level by which we know that how the vacuole divides and why the vacuole is important. This can also be utilized to design a vaccine, because you know, if you are constructing any vaccine strain which is giving a potential antigen to be delivered into the DC, you want to keep it protected for quite some time. You don't want antigen to come into the system and then it is getting cleared. So this is a really good system, that you get it protected in a vacuole for some time, and when you want it to be delivered, you puncture it by certain pore-forming toxin and get the molecule out into the system at the right time. So this is a very important tool also. As far as the pathogenicity is concerned, it can be, the salmonella goes into the epithelial cells, it survives because of one kind of a transporter. Epithelial cells are very rich source of antimicrobial peptide, the defensin categories. But remember, salmonella is not freely moving in the cytosol. It's always covered by its vacuole or its niche. So nothing can touch it. If it is in a macrophages, there is a big burst of reactive oxygen species and nitrogen species, and there also it is protected because of this covering. In the dendritic cells here, what they do is they will inhibit the antigen presentation, again because it is covered. So you know, one can do a very cool experiment. I will tell you that if you make a pore into this vacuolar membrane, by expressing a pore forming toxin, it will come out. When it comes out, everything changes, everything goes haywire. So much, so all my colleagues, my PhD colleagues who started, every time they did this discovery, they were baffled by the fact that indeed this had such a multi-spectrum behavior of getting all the cell type. How a bacteria, when it switches from the epithelial cells to macrophages to dendritic cell, can sense. And I know that you all know why it can sense. They sense because of a very important signaling mechanism, and that signaling mechanism is nothing but two-component signaling system. This system is also harnessed a lot in the biotechnology platform. You can make an amazing circuit out of this two-component system, because one is a sensor kinase and one is a response regulator. So this module works for you to express any gene that you would like to do it. So you can modulate this two-component system also. So with all of this, the salmonella became a very successful pathogen. And not only that, they became a successful pathogen. So this is a cell, macrophages. You can do again the experiments in the normal cells like macrophages, raw 264.7 cells. The green are the bacteria here. So you can infect them and you see that they are all within one cell. When they infect the macrophages, they totally destroy the lysosome. So lysosome is also one of the important organelle for affecting the bacteria, but bacteria has its own strategy. So you see what is a tug of war? You put an assault, they also put an assault. We have a tons of lysosome in our cell. So the bacteria, the moment it comes, it totally stops the lysosomal biogenesis. This is also one of the important tool to understand how, see, lysosomes are important organelles not only for bacterial infections, but for virus and including the metabolic disorders like cancer. There are lysosomal storage disorders. So for all of that, this is again a nice model system to understand. But as I know that your one of your colleague was talking about the safety issues, I would take it with a pinch of salt that you do not have a permission to work with salmonella in any lab until and unless it is a IBSC certified lab. Yeah? So even if you want to do any experiment, it would be good to approach a lab which is certified, which has got its IBSC clearance. But if you want to work with a mutant strain, then you can work anytime because they are as good as E. coli. They are absolutely harmless, so no problem. Yeah, so you can mark it. Look at this beautiful salmonella which is residing within a vacuole. So these are the vacuole marker. There are 40 different protein which make up this beautiful vacuole around. So this is one of the marker. You see that this is single indicate. So we talked to our physicists, mathematicians, that's what I said, no microbiology without these three subjects. All of them, they told us one very important point. This is not a very, simple process of the bacterial division happening and the suction force is getting your outer membrane sucked inside. No, this is a very important process which needs to have some metabolic activities by bacteria. And that is indeed true. You look at the dividing bacteria and what gets recruited to the site of the division is endoplasmic reticulum. So the endoplasmic reticulum marks the site of the division and then it does the so-called bacterial vacuolar fission. So just like your mitochondrial fission, they wrap it around. Now we have much more word on that how do they wrap it around and how do they get the fission done, what brings in. So there is a bacterial protein which brings in. So that is how they do this fission. So the moment you perturb this activity, all the salmonella, they come in one big vacuole and then they die. So single bacterium per vacuole is indeed a way by which salmonella progresses its life in pathogenicity. So the perturbation, so if you are thinking of an intervention strategy, how many of you here are working on something to do with intervention strategies of microbe, any AIIM project? Thinking about how do you intervene or how do you stop the infectious disease? Nobody in this group? Okay, that's fine. If somebody is thinking about the intervention strategy, it is good not to look into how will you kill the bacteria. It is good to have a little bit of a host directed approach where you will not allow this vacuole to be dividing. If you do not allow the vacuole to divide, they all end up in one vacuole and that kills the entire population of bacteria. So now you know that how beautiful is their studio apartment and why do they love to be in their studio apartment. Less competition for the food, less competition with any of the defense molecule, and each one of them, though they are separated by a cover, they are communicating with each other. That brings to this quorum sensing. Within, how do they do this quorum sensing and how do they communicate with each other within the cell, though they are separated by a system. And we know now from our own evidence that they happen. So I think my time is up now. It's still 12, no? My talk? Yeah? Okay, so colleagues, maybe more stories sometime later. What I'll do is I will quickly just get to my group now. So this is my group and they are all, so I always say that the legends are in a vacuole and then they will split apart and they go to and make their own different direction. So this project was started by my colleague Dr. Sandeep, who was an MBBS graduate with a great interest in, again, physics, chemistry, biology. Like any other, parents, his parent also only wanted him to become a doctor and he became, but a crucial time came when he got his seat in MD. So when that time came, he quickly gave his ISC entrance. I'm talking about 2004, and he did not tell his parents and he joined my lab. So the parents came to know that he is not opting for MD. As usual, many things happened. Then the parents came, I talked to them. He did very well. He is currently associate professor in Department of Biochemistry in IISc, working on an amazing problem of, looking into this something called as in the translation and the entire mechanism. So he started the lab, followed by all of them. So the lab has grown very big, and actually the collaborators, what science gives you, and that is what you are all here, I think, that this particular AIIM meeting with all of you around from different places, different ICEs, makes you very good collaborators. So we know each other, all of us. You can approach us anytime for anything that you would like to do, and any idea that you would like to vet or you would like to have our opinion. So that is what IISc gives us, and I would definitely like to acknowledge everybody in the team, of course IISc, and yes, the last year's, 2 years back's AIIM team, they came up with a beautiful book that is Resistance Tale from the Post-Antibiotic World. This was a 2019 batch of the AIIM team, and I was definitely mentoring them. I was very proud that they came up with the book. That book is released by the IISc Press, and I wrote a very small poem for that book. Thank you very much, colleagues, and I'm open to questions. Yeah.
Are there any questions? We'll pass the mic around in case anyone would like to ask anything.
Yes, yes. Yeah.
You told, is it audible? Yeah. You told that.
It's not working. Can you just be a little
Hello.
louder? Yeah, yeah, yeah.
You told that even after the typhoid infection
Yes.
is like there are no microorganisms, but still there are after effects such like mood swings and depression. How does that work?
How does that happen?
Yeah.
So I'll tell you. Now currently that is what we are working on, that because we realize that there are too many cases now of meningitis. So what happens is Salmonella, though it gets through the fecal-oral route and it spreads across, it has a certain virulence factor which makes it to somehow destabilize the blood-brain barrier. Now the Salmonella also gets into the brain. So we are seeing it in the medulla, we are seeing it in the cortex. When we give antibiotic, it gets cured. But when we are doing a lot of these maze tests and other memory tests, etc., of course we have a long way to go now. We are seeing that the retention, the memory retention is lost. When the bacteria is lost, is cleared by antibiotic, for many days, still they are not able to come back to the original wild type mouse. So we are looking into a lot of these PV interneurons to understand that what has gone wrong and whether there is irreparable damage that has happened to certain sections. So yeah, it is indeed very interesting to see that happens. But we will also be interesting to know that what is the load of the bacteria that is required to give this irreparable damage. Maybe a low dose of the bacteria may not be able to do so, but if it has become meningitic and if the dose was very high, then those damages are irreparable damages. So the best part is do not allow this bug to get inside you. There is no reason, that is the only reason why the WHO after these many years have actually put along with escape pathogen this bug in the high priority pathogen list. There is no vaccine too, right? So we are at threat at the moment. Yes.
Thank you, ma'am.
Yeah.
What is the mechanism for the asymptomatic transmission of
Asymptomatic transmission?
Yes.
Good question, very good question, and Mary Mallon was really
Yes.
kind of isolated because of that. So 1% of the people who are infected, they become asymptomatic carrier. Now from 1% those days, now it has become little less. What they do is, instead of clearing off the salmonella harbors and stays in gallbladder, right? So you can imagine, they have a mechanism to sustain and survive in this very toxic bile juice, which is stored in the gallbladder. Now with every squirt of bile that comes out, this salmonella comes out into the system and they spread it. But your question is very valid. Why they are asymptomatic? Then they should harm the host also. Now this takes us back to the host immunity and certain host immune parameters which does not allow the bacteria to manifest its symptoms. And that is in the 1% of these healthy carriers. So you call it as asymptomatic or healthy carriers. And we have a mouse model for that, but I think there is too much yet to be known. Yeah.
Thanks.
Yes.
Ma'am, you told that the bacteria are able to communicate with each other
Yes.
when they're in different vacuoles.
Yeah.
So how are they able to do so?
How are they able to do so? And till so far it was not known, because there are very few examples of a bacteria which is in a vacuole, and that too in a tight fitting vacuole. You can have a spacious vacuole like Legionella, it stays in a vacuole but big vacuole. Now this we are talking about like this, tight fitting vacuoles, right? Now what we have shown is that the so-called quorum molecule, which is belonging to the category of the auto-inducer, AI molecule. Now this AI molecule is formed and it crosses this vacuolar membrane. And from here, this AI molecule can also enter this vacuolar membrane and signal this bacteria. So this is how you have a very beautiful kind of a balance between the AI molecule which is outside and the AI molecule which is inside the system. Now the individual bacteria getting their own signaling by the AI molecule makes this bacteria work in a consortium. So always, you know, when you look into the sections, be it an organ section or a cell section, you will always see that these bacteria stay together. You will never see, suppose this is a macrophage, you will this is a nucleus. One is here in its own vacuole, one is here, never. They are always like this in a consortium. So this nearness, and we are trying to actually understand what is the distance and why do you do that? Can you perturb this mechanism and still see whether the pathogenicity is retained? Yeah, so intercellular quorum is an emerging field, and it is only because of this that the successful pathogenesis is caused. Though we are studying Salmonella as a model system, the same will happen with everything, be it Acinetobacter or Klebsiella or Staphylococcus aureus. Yeah. Yes.
Ma'am?
Yeah, please go on.
Ma'am, I had a doubt.
Yeah.
I think I attended a talk recently in that MCB seminar hall.
Yes, yes.
So there you said that if you did some certain mutation,
Correct.
then the salmonella can exit the vacuole.
Correct.
My question is, will it still have the same pathogenicity once it exits the vacuole?
Yeah, so very good question, but a difficult answer at this very restricted window frame. Whenever, say for example, this is in a vacuole and you express a protein from Listeria, this is a beautiful pore-forming toxin. You can genetically engineer your salmonella and you will see that when it expresses LLO, this will quit and come into the cytosol. Now this particular bacteria, they do very well, but it requires that some time frame for it to stay in the vacuole. So staying in your own home for some time is very important. You know, it's so interesting to see that this LLO, bacteria with all the gene, but you express LLO, they come out, they do very well. But we have a mutation in a gene which is in this AIIM system. This is outer membrane porine. Now if you make a mutation in the AIIM, it quits the vacuole much faster than the LLO. The transit time for the vacuole must be 15 to 20 minutes. But AIIMP mutant makes the bacteria to come out in 5 minutes. They come out and they die. So how much time you spend in your home? Why? Because you know this vacuolar environment is acidic. And in this acidic environment, they secrete their secretion system needle, which is encoded by pathogenicity ILN2. And they all belong to this type 3 secretion system. These are the beautiful molecular injections that I was talking about. If you do not give this transit time, they will not make this needle. If they cannot make the needle, the virulence proteins will not be expressed, so they will die. The moment you have this, they will die, but if you have LLO, they will take some time to express LLO and come. By the time the bacteria gears up. Yes. Yeah, the bacteria in the cytosol, good question. This bacteria in the cytosol continue to divide, and suppose if you have a neighboring cell here, this bacteria, okay, now this is an important question which we have not studied. How many rounds of replication can this bacteria undergo? No, you mean the one which quit the vacuole? Okay, now. Yeah. No, I will tell you two situations. The first situation, the bacteria is within the vacuole. When they divide, the vacuole also divide, and they will be always within the vacuole, always. There is not a phase in this stage that they will come into the vacuole, because vacuole cannot be formed de novo. The first vacuole is formed from this plasma membrane. You know, the bacteria comes, there is a dent, the bacteria comes it pinches off, and this is the so-called naive vacuole. From then on, it is always vacuolar. Now, if you somehow, due to whatever reason, they quit it, they make to quit, then they will be within a cytosol. If it is this kind of condition where you allow them to stay for some time and quit, they are geared up. So, they will go on dividing. But if you make a mutation in a gene in bacteria like OMP-A, they will quit much faster, not 15 minutes but 5 minutes. They come, they are not geared up enough. Then they will divide one time and then they will die. This is clear? Yeah. Yes.
Ma'am, I have a question.
Yes, please. Yeah. Yes. Correct. Yeah, that is actually the one which the vaccine industry is trying to utilize, the secretion system and the delivery system. There was an amazing paper in Science which talked about how a viral autologous antigen is delivered through this system. Yeah? And yes, you can tune it in a way that you have your bacteria engineer with that particular antigen. Now this is in a vacuole and they are here. You also tune it in a beautiful way by which they can make their secretion system apparatus right to deliver the protein. Now, if it is a viral vaccine, in fact, you need not leave the vacuole either. This will allow your antigen to be secreted by this molecular injection needle right into the cytosol, and suppose it is a dendritic cell, they are going to do the antigen presentation. But if you have a certain other kind of vaccines, suppose they are displayed on the surface of bacteria, antigens which are displayed on the surface of bacteria. In that condition, it would be nice to cleave it and allow the bacteria to come with the antigen displaced on the surface, so that now this whole complex can be recognized by the antigen presenting cells. Yes, you can use it and tune it and engineer it to give a very efficient antibody or antigen response. Yes. Last question? Okay.
Yeah.
Yes.
Ma'am, I have a question.
Yeah.
So you have told about different cell types
Yeah.
of pathogenicity of the
Correct.
bacteria. So in the vacuole, like when the organism in the vacuole, does it regulate the gene expression patterns of the particular host cell?
Oh, of course.
So,
Plethora. You know, it's like a drop in the ocean. You put a pebble, you see ripples. You put a bacteria, you see a ripple. What is not touched? Lipid biosynthesis, carbohydrate metabolism, protein metabolism, every single thing. So now it's coming in different area. You have a TCA in bacteria, you have a TCA of the host, they are interplaying with each other. The same is with lipid.
So basically they are not cell-specific genes, they are like housekeeping genes, so that like all the cell types are like getting affected. So
Not the house specific. See, for example, if it is in dendritic cells, they will perturb the antigen presentation machinery. If they are in the epithelial cells, sometimes they will downregulate the antimicrobial producing genes. If it is in macrophages, they downregulate the ROS machinery. So like that.
it's like changing, something must be common, like we can't find it, like which is common, like on all different cell types, which is a common mechanism, this
There is
particular.
no common mechanism. That is the beauty. Yes, yes.
Okay, any other question can be taken outside the hall.
Yes.
Thank you very much, Deepshika ma'am.
Thank you.
Now we'll honor her with a small present.
We'll just close the file and then.
Yes? Okay, maybe I'll just wait a minute before you all settle down because I see quite a bit of action. Because I have a question for you guys. How many of you here are non-biologists? How many of you, okay, very good. That's a large number because I am a non-biologist. How many of you here are non-engineers? Okay. How many of you here are physicists? Mathematicians, computer scientists? Okay, so there's a fair mix, okay? And this is of course the meeting of the AIIM, and I believe it stands for genetically engineered something, right? Sorry, and the origins of all of this is in this field called synthetic biology, correct? And you know, I didn't know too much about synthetic biology till a few years ago, so I dug myself into some history of this field. And it's around early 2000s is when the first AIIM meeting happened at MIT, okay? And I also saw a list of all the things that are considered as milestones in this field, and they are all broadly related to either engineering aspects, as they write here. In fact, you know, there are some cultural milestones. What do we mean by cultural milestones? I think they mean some, you know, some way, some philosophical way of looking at this thing. Of, you know, Monod and Jacob introduced this idea of gene regulation. Yeah, that's a cultural innovation. But most other things are circuit engineering, metabolic engineering, therapeutic applications, whole genome engineering, so on and so forth. And I also got to see some of the applications that some of the AIIM teams are here doing. And they broadly fall into this class of applications, yeah, for health, or for climate change, etc., etc. And here today I am come here with a manifesto to say let's try and look at it, synthetic biology, from a slightly different kind of way. And I'll tell you what in a second. So this was roughly around, you know, the early 2010s. Of course, the field continued on, and I saw even more milestones, and they're all along these very similar trajectories, ending all the way in 2019 about carbon fixation, climate change applications, and so on and so forth. Okay. And since I never got myself into this field of synthetic biology, I said, what is happening now? So I just randomly picked some university, Wageningen University, and I looked at their synthetic biology department, and this is what they have. Synthetic biology is fed by all these various things. Okay? And it's not surprising when I looked at the previous two lists, it was not surprising for me where things like agriculture came in, or microbiology, etc. came in, where something like chemistry came in. It was also obvious where things like engineering or computer science came in, because there was circuit engineering in there and all. But there's this thing of physics. And in none of the examples in the past, I saw physics. And this bothered me. Where is physics entering synthetic biology? And it occurred to me that, okay, let's first look at how, you know, these are classic examples that we have just looked at right now, that I can build a circuit, I can regulate something, I can design something in order to be an amplifier or an oscillator or a sensor, and so on and so forth. And this is all language from engineering sciences or the computer science. I can build a logic gate into it, a computer science. But where is the physics? Yeah, how is physics coming into this whole game? And I didn't see any actually. Okay? And here today I am standing in front of you with a manifesto and a proposal to say that there's a very interesting way to bring in physics into this game. How do we do this? Does any of you know this gentleman? Any ideas who this gentleman is? No? No? Maybe slightly, he's a scientist who has had a deep impact, of course, on biology, and maybe slightly before your times of knowing who he might be. His name is Carl Woese. Carl Woese, he's a physicist by training who turned into a biologist, right? And he was the identifier of the third branch of life, the archaea. And I'm sure you know how he did it, using the 16S and all that stuff, right? So Carl Woese is a physicist by training, and there's a remarkable interview of his in this journal called Current Biology. And I've picked for us to read 3 questions and 3 answers that Carl Woese has given in this interview. First, so he's just saying, you know, the previous question was, how did you enter this field of biology, being a physicist? And he's given an answer. And he's said some things which certainly to the interviewer sounds heady. So the interviewer is saying, you're saying some pretty heady stuff here. First, what is wrong with biology becoming bioengineering? Clearly he has said something to the effect of biology is becoming engineering. Remember, this is what I also just said now, that the synthetic biology is engineering. So in all of this language, you might replace biology with me saying synthetic biology, and you'll see what I mean. So this guy is asking, what is wrong with it being biology? Carl Woese is saying there's nothing wrong with bioengineering per se, but it is wrong when bioengineering comes to define biology. And I think this is true of synthetic biology as well. I think it's a danger that application in engineering come to define synthetic biology. Physicists understood this a long time ago. Physics formally split into 2 allied disciplines. One became physics and the other became engineering. Okay? And he goes on to say that biology needs this. Some equivalent of physics, some equivalent of engineering. Okay? The broad goal of science, he says, on the one hand, science is society's servant. Everything that you guys have put together as, you know, it's a servant. It's saying, society is saying, I have this need, solve my problem for me. And as very good servants, we are solving or tackling those problems, climate change, health, etc., etc., etc. But science has a very important other role. What is that? It is society's teacher. Okay, it's not simply saying, society is saying, give me something to improve agriculture or for me to improve my health. I'm also, as a scientist, going to teach you of how to place yourself in this universe, in this world, right? And he goes on to say, it is this latter function of science as society's teacher, which is effectively missing today. And this is sort of coming to define biology and also synthetic biology, in my opinion. Right, so he goes on to ask, so does biology need to go all the way back before all of this came and start in the early ages? He says, no, biology has its physics. It has its equivalent of modern physics, and he says that physics is actually emergent self-organization. And he says, here is the physics for a 21st century biology. Okay? And I'm going to try and give you a few examples of what we are trying to do using such a perspective in trying to define synthetic biology from this physics perspective. The last question, how important is the relationship between biology and society? You can replace biology with synthetic biology. And he says it is all important. It's a matter of deep concern for all biologists, philosophers of science, and political leaders. And I don't see it being taken very seriously by any of them. The intersection between biology and society is mutually self-shaping, mutually shaping. Yet the interaction now that exists between them is nothing short of intimacy. He's saying it's trivial. What is he saying? Biology today is conceptually weak. I don't want to trivialize the synthetic biology that exists today, but coming from this physics perspective, I am saying that it's conceptually weak in this sense. Biology today, it's putty in society's hands. It is surrendering to servitude. It's delighting in becoming society's genie. Somebody is saying, hey guys, there's a problem for me in agriculture, and we are saying, can I design a better circuit to solve this problem in agriculture? Which is fine. I'm not saying it's a bad thing, but it should not become the only thing, right? So, and society will accordingly ask for it more. Make man the medical miracle, make it, utopian agriculture, etc., etc. And he ends saying, biology, know thyself. Know yourself. Uphold your end of the bargain with society, provided the counsel. In other words, he's saying, become society's teacher. And he provides a route for it. Mankind needs to appreciate itself as a part of the evolutionary flow of life. Okay? And this is where I'm going to pick up the thread and use it to come at synthetic biology from a physics perspective. And it's through this lens of evolution is what I'm going to say. Okay? So let's start off from this branch, physics. And I've picked a beautiful review article from another stalwart here in IISc, Sriram Ramaswamy in the physics department. And this is a review article about this field of active matter. Right? It's a field of physics. But look at the first line he writes. Physics enters biology in two broad and overlapping areas, information and mechanics. And I'll try and unravel these things for you a little bit. Mechanics, and this review is about mechanics, statistical or otherwise, of living matter. Okay? When I say physics to you guys, what's one of the first things that comes to mind? Sorry? Mechanics, what else? Space. Say something. Stars, pendulum, states of liquids, gases, solids, states of matter, water, quantum, yes, exactly. Quantum, all of this has to deal with matter and material, okay? So matter and material is what physics deals with, and we have classified matter and material into gases, liquids, solids, and so on. But there are also other interesting classes of materials called liquid crystals. Have any of you heard of liquid crystals? The liquid crystal TV display is the same thing, except these materials exhibit very interesting properties because the molecules that comprise those materials are like rods, unlike spheres. Okay? So much so that you can have other states, unlike gases, liquids, and crystals, you can have mixtures of liquids and crystals in these materials. Okay? So look at this. This is a so-called nematic state. It's a bunch of rods which are aligned more or less in the same direction. Okay? This is called a nematic liquid crystal. So what does a physicist see when he sees a school of fish? I am seeing a nematic phase over there. Okay? So I am viewing biology from a slightly different, twisted way. Okay? As a physicist, I am viewing that in a wonky way. I am viewing them as rod-like molecules. So now, as a physicist, I know a lot about this kind of material because I've studied it in the lab. I've written equations for it. I've understood it, how does it behave when I apply an electric field to it, etc., etc., etc. Now I've written down theories for that. Can I do the same for this? You will say, come on, this has a brain, this has eyes, this has all kinds of things. How can you just turn them into simple rods? But it turns out that we can actually do this to a very good extent. Okay? And this is what has come to define this field of active matter, so to speak. Using the ideas of materials that we know from studying physics in the lab and applying it to biological systems. Okay? And in fact, this sort of thinking of this active matter can spread across scales. So what you're seeing in this slide, so on one hand, I'm showing you an experiment which is from the cytoplasmic extract of frog embryos. In the middle is a movie of the development of the frog embryo. In the last is this flocking of tadpoles. Okay? But it turns out that when we write the equations, governing equations for phenomena across these scales, using the ideas that I just told you, it turns out that the equations are all the same. This is quite remarkable and quite beautiful actually, that my thinking of treating or simplifying concepts across these scales can work across. So there's some idea of universality that physics is bringing in, which helps me understand biological phenomena. And this is what is this field of active matter. And this field of active matter, what it has led to now, so for example in my lab and many other labs, is synthetic versions of those phenomena, right? So for example, here is an experiment that we did with droplets of oil. in water and some soap. That's all it is. Okay? And you see that it displays some remarkable banding phenomena. Here is another beautiful experiment from physics department in IISc. This is a bunch of pins, steel pins, which are on a shaker and they're being shaken up and down. And you see that they exhibit this beautiful flocking behavior. Okay? Here are 3 movies. Oil droplets, bacteria, starfish embryo. You are an alien and you're watching these movies. You have no idea what is biology. Can you tell me which is biology and which is not? It's hard, right? But the question is this a synthetic biology? Why not? It looks like biology. It behaves like biology. It's showing properties like biology. Why is this not a synthetic biology? It is a synthetic biology. It's a different biology. Like from my point of view, it's different. You might say, what is it useful for? But I'm telling you, that's not the aim with which I started. I'm not looking for use. I'm saying, what is it telling me about the universe? What is it telling us about our place in how to think about the universe? Right? That was my initial aim. And this is telling us that even inanimate objects under certain conditions can provide you with dynamics which look like living systems. This is quite remarkable. But I would still hesitate to call this a biology for the following reason. The same reason why I wouldn't call a car biology. Why? Because I think fundamental to biology is this aspect of being able to produce another of its kind. Reproduction, growth, division. What does that growth and division really lead to? It's not merely about making more copies of itself. It leads to this very important process of evolution. Why? Because you make a copy, the copy can be faulty, and then you have competition or whatever else, and then you can get an explosion of complexity. Therefore, I would hesitate to call this still a biology, right? Because a biology is comprised, a single unit would be a cell, and the dream of every cell, as Mono said, is to become another cell. And I would say this is the core in some sense of biology. So if I were to come at synthetic biology from a physics perspective, I not only want to mimic some features like that, but I want to include this growth and replication in a fundamental way, or evolution in a fundamental way. Okay? And like I said, just going back to Bose's thoughts, I need to include this evolutionary flow back into my thinking of the physics of synthetic biology in a deep way. Okay? And this is where I want to point out to all of you guys that evolution is the ultimate engineer. Okay? We might come up with designs of circuits, what have you, but evolution has been doing this remarkably well for millennia. As a tinkerer, and in this process of evolution, physics enters naturally. You don't need to put it in, you don't need to take care of it. And I'll give you couple of examples of this. So my lab at NCBS takes this viewpoint. So we want to take, this field of active matter I told you. What it's giving you is, it's giving you mechanics, because it's giving you things that are moving, that are interacting, that are clumping, grouping, forming. So we are trying to combine that aspect together with this aspect of replication, assembly, and so on, basically evolution, and combining these two and asking, can I drive things into what I call a synthetic biology from a physical perspective? Okay? So this one we are doing from 2 points of view. One is like these oil droplets, to take systems which a priori are non-living and try and get them to become more and more lifelike, move, grow, divide, etc., etc. And the other is to take biological objects, cells, and put them through evolution and ask what can happen of them. And I'm going to give you one example of this kind, and very briefly allude to this in the very end. Okay? So the example I'm going to tell you about is, how can I evolve large size or multicellularity from small size or single cells? Okay, this is the question I'm posing. Why do you want to do this? What is the use? Well, I want to learn what biology can do. Okay? Okay, so here's the experiment. So we start off with single cells of yeast grown in the lab, and the selection protocol is as follows. You guys know that you can grow yeast in liquid culture, shaken liquid culture. So what we grow that in shaken liquid culture, and then we put that liquid culture down on the desk and let it settle under gravity for, let's say, half an hour. And then we pick from the bottom. So if you're able to settle to the bottom in half an hour, you survive. So if you're bigger, you have a selection advantage, right? And therefore what we saw after a few months of this, just doing this, nothing more, is objects of this sort. These are single cells which have divided but have not separated from each other. You might say, this is what a colony does. What is so special about these guys? What did you achieve by wasting 3 months? I could have got this by growing it as a colony. What I want to tell you is, this is not simply a colony. In the meantime, by the way, we have these sort of clumps which are about half a centimeter in size, massive. Remember, I've come from an object which is 5 microns in size to something which is half a centimeter in size. Why is it simply not a colony? Instead of giving it 40 minutes to settle, I can give it only 5 minutes, which means that I have to become even bigger, right? Unlike a colony, these clumps do something else. They divide into clumps. These are the movies that you're seeing over here. The scale bar is the same across movies. So you see that there are smaller clumps and bigger clumps. If I give it only 5 minutes, it needs to become bigger to settle, so it will end up there. If I can give it 1 hour, it will end up here. Okay? But here's the cool thing. If I take a single cell from this cluster, it will grow to that size and then divide. If I take a single cell from this cluster, it will grow to that size. And now this is very interesting because it's not simply a colony. Right? This has gone beyond. And if I take this guy and evolve it under 2 different settling regimes, it will end up with 2 different sizes. Okay? What, in effect, what we have created is a primitive organism. Does this exist in the wild? Does it exist in the, I don't care, because I can create it in the lab. This is in my point of view, this is a synthetic organism which I have been able to create simply by using evolution as a tool. Now where does physics come in? When this organism gets bigger, mechanics comes in two very fundamental ways. Some of you might know this argument by Galileo. He asked, can I have a mouse the size of an elephant? And he reasoned no. Why? Because he said everything has to scale, and if everything, the bones have to scale, everything, so, but they're not strong enough to support the weight. So this thing has become half a centimeter big. How is it supporting its own weight? It turns out that the shape of the single cell has also evolved to become rod-like, and therefore effectively change forces and make it more rigid. And another mechanics reason comes in when organisms get big. How do I feed myself? Yeast, as you know, unlike bacteria, cannot swim. It cannot stir up the ambient medium, etc., etc. Correct? So if I'm beyond a certain size, diffusion will not feed me. My feeding requirements grow faster than diffusion can bring in nutrients. So what can this huge thing do? Not grow. Not grow. It's just big, but I don't grow. But what do we see in our experiment? We see that this guy, even at about half a centimeter in size, it's growing exponentially in size. What does exponentially growing tell you? It's growing everywhere. How is it doing this? If I now remove the liquid medium around it but put it on agar, this exponential growth goes away. It becomes linear. It's telling you that, hey, I need liquid around me. Why? Because it can support a flow Physics comes in. Physics comes in a very interesting way. So is this thing making flows? Yes. So here's the organism, and these are just tracer particles which we put on the outside. And you see that it's mixing up the fluid. How is it doing? This must blow your mind, because this is a little bit like saying, I'm going to take an ant and grow it bigger and bigger and bigger and bigger, and suddenly it starts to fly. It's analogous to that. Remember, single cells of yeast don't have flagella. They don't have any mechanism to mix up fluid, but somehow this big guy is able to stir up fluid. Okay? And if I look from the side, so this is this organism, so you see this flow, it's rising as a plume. It's mixing it up. Have you seen this sort of flow somewhere in the ocean? Sponges. Are we evolving a sponge? I don't know. Are we evolving something else? I don't know, but it's clearly a synthetic biology, because it's allowed by the laws of physics, it's come from evolution, it's taking advantage of new physics in order to feed itself, right? But I don't care that it exists in the wild or not, because I'm learning something new about biology. It's telling me something about evolutionary tracks. All right, I won't go into the details. It turns out that they need to be alive and metabolizing to generate these flows. The flows are sensitive to gravity, so they're driven by density. We can talk about the mechanism later, but here's another cool thing. When someone tells you, why do birds flock? Why do birds flock? What's the reason for flocking of birds? Sorry? To facilitate collective flying. Why do fish school? Huh? More secure. How can we check these things? How can we? This is a story. I might say the reason for them is to look beautiful. How do we check it? It's a statement, right? Unless we are able to perform an experiment in the wild, we are not able to verify that statement. But here is something very interesting. This object, this organism is producing a flow. And because it's producing a flow, physics, nothing else, no biology, no ecology, nothing. Physics dictates that they will aggregate. Okay? So here you are seeing on a Petri dish, guys which are dead, guys which are alive. You will see that the guys which are alive at different sizes, they're all aggregating into clumps. Physics is pushing them. It has no option. It's like if I put a roll of ball on a hill, it has to go down. Physics dictates that. The ball doesn't have a mind. These guys don't have a biological use for this, but physics pushes them there. Okay? And another surprise, if I look closely into this movie, they've all aggregated, but look closely at one guy. Shooting off. Shooting off. Okay? They are suddenly motile. Look at that guy. He's just moving around, walking. What I want to point out to you is the following fact. What we did, the evolution was simply to grow them bigger, nothing else. They're producing flows for free. They were able to move for free. I didn't impose by selection the ability to move. I want to point out something to you as synthetic biologists and designers over here. When you design a particular feature, that's all you design for. That's all you get. But look at this. I selected for bigger size. I'm getting flows. I'm getting motility. What else I'm getting, I'm not telling you, but I'm also getting the fact that this object is turning into a vada. There's a hole that forms in them. It's changing shape. All this is coming for free. Why? Because I've paid attention simply to evolution and the fact that physics is doing something interesting over here. Okay? Finally, I will end with this one example that I said on the other branch, taking synthetic stuff, right? So we are trying to construct a synthetic cell from a primordial soup. And this primordial soup, again, hold your horses, it consists simply of salts. There are no lipids, there's nothing. Simple, small molecule salts. common salt, some formaldehyde, magnesium, potassium salts, there's a molybdenum salt, and we get these things. And we get these things, and these are, you can see that they are growing in size. They're about a micron. If I take a section inside them, they look like that. If you were an alien, I showed you that, you'll say, ah, there's a nucleus and there's a ER and there's a Golgi or whatever else. I don't know. What we know so far about these or these creatures or these objects, they're compartments, they're doing some metabolism, starting from simple salts. They're doing all of this stuff, they're growing, they're also motile. And I want to humbly submit to you that this is also a synthetic biology, but it comes from a different perspective, right? And it's coming from a perspective not of engineering, but a different worldview on this whole thing, and perhaps that is useful and interesting, okay? And if you're interested in any of this, so Nishant and Nayan are the two PhD students. Nishant is doing this yeast stuff, Nayan is doing this latter stuff. I'm at this place called the Simon Center for the Study of Living Machines, and we have a new PhD program on the physics of life. Look it up, look it up. Thank you.
If there are any questions, we'll be happy to take them.
Hello. Very nice presentation, sir. And I have fundamentally two questions. One question is that usually whenever people talk about biological experiments, there's a lot of emphasis towards this so-called randomness. And usually biology is taught, like experiments are taught to be something like a black box where you just set it up and you just see the results while you don't really understand what's happening in the middle. So do you think if we know more about the physics of all of this, then that black box might somehow become something very tunable in the future?
Yeah, that's a very good question. And it's a black box, not because we don't know anything about it, but it's black because it's so dense that it looks opaque to us. There is so much going on, okay? That's the reason why biology is hard. But an approach like this sort of breaks that process down in interesting ways. And I'll tell you why. It's not that you get rid of all the complexity, but you break it down slightly, because you're right, one, there's a physics or a mechanics perspective from which I can say, hey, what has changed, which I can understand simply from a quantitative perspective or by writing down a bunch of equations which I can analyze and get out. But in this evolution process also, by the way, I can keep track of every mutant that has happened. And I can know even at the molecular level what are the changes that are going on. And in fact, we know the reason for why that thing doesn't break. It's because there is a mutation in the chitinase, and the chitinase doesn't break down the cell wall material, and therefore the cells don't separate, right? If I, as a design problem giver, gave you this problem saying, can you design this thing into this yeast, you'll probably come up with that solution, but not maybe a priori to say that it will grow into this large multicellular thing, it will generate flows, it will no, right? Therefore, there are things which are emergent, which we understand only later, but there are features which you can understand in a systematic way as well.
Thank you. And the second question is, usually I've noticed that a lot of proteins start misfolding and aggregating on the mutations and stuff. So since you were talking about aggregation and related to basically directly from physical laws, so could that play a role? Like, why would
sure,
a mutation?
This is also the reason why a lot of physicists, my lab included, are thinking about protein aggregations, because that is also akin to phase separations in physics, okay? And whether, and from this viewpoint, the mutations that you're talking about just give rise to a different kind of polymer. That's all. I don't need to ascribe any biological function or feature to it a priori, right? Thinking of it as a physicist, I have a polymer, which is your protein, if I change the nature of that polymer, the alphabets along that polymer, of course it will have a different physical and chemical property. Therefore, it will have a different way of aggregating, it will have a different viscosity, etc., etc. Now, biology comes in because it can take advantage of that and adapt, co-opt that into function. Now, this flows has come for physics reasons. Biology can take advantage of that. If I run selection on that, it may get incorporated into the genome. The motility similarly, and so on. But initially the processes are happening simply because of physics.
I think you answered my question. My question was whether it is purely a physical process or the biological organism takes some internal mechanism in order to cause the flows, and what the mechanism is.
Right. So the mechanism I haven't told you at all is because this organism takes up sugar. It's the nutrient, right? And when you take up sugar, you know that sugar water is denser than water. So I have a lighter fluid below a denser fluid. So the lighter fluid has to rise. Once it rises, there's a flow. It's simple physics. But then you might say, but if I now, I haven't asked this thing in my selection regime to respond to that at all, but now I can say, can you make a better flow? Then maybe it changes something in its metabolism and so on, right?
Thank you for the session. It was a very new way of looking at biology. So my question was, you said that similar equations hold true for everything from cytoplasmic to your tadpoles. So my question is, why is that? Does it have to do with now physical systems take the path of least resistance and hence they fall a certain way? Is it that biology has leveraged that mechanism and does the most efficient way? Is that why the equations hold, or?
Yeah. That's a very good point. And I must clarify what I mean by equations hold. First of all, what are the equations for? Okay. So the equations are simply to ask, the equations are simply to say, if I have a molecule or an object and it produces a force and I put a bunch of them together and the forces interact, what organization do I get? That is what I'm trying to describe. Okay. So here there are a bunch of microtubules acting and they aggregate into a cluster. Here there are a bunch of force generations in the cells, etc., which reshape it. Here they produce forces and swim, and so ultimately it's about mechanics, force propagation, and the collective behavior. And that is what I can describe using these equations. And turns out that the equations don't care whether they're at this scale or that scale. It's not a coincidence. I wouldn't say it's a coincidence. I would just say that we are trying to describe a mechanical process, okay? And therefore, now that we know what the mechanics are, that force development, force generation here versus there and so on, there is in fact a deep reason for them all to be all similar. So, let me give you the following example. An apple falls down due to gravity, and the planets also interact due to gravity. There's this notion of universality in physics across scales. It's the same thing that's going on over here. The same equations produce the same behavior. The scales really change a little bit, not much.
Okay, if you have any further questions.
If you have any further questions, please reach out to me. My email is my first name @ncbs.res.in. I'm happy to do that.
We'll also share the basic contact details of all the speakers after the event is over, so you can always get in touch with them. Yeah. Thank you so much, Dr. Shashi. Please, sir, can you stay on stage for it? So, just a small token of our Thank you, sir.
Can you hear me at the back? Yeah? Awesome. No, I stand between you and lunch, but then you all also sit between me and my lunch. So we'll try to keep each other, we'll bring each other as close to lunch as we can. Cooperate in this process, and cooperation therefore will be the theme for this short talk that I'll try to put across in the next 15 minutes. I'll be sympathetic. So, given the abbreviation, therefore, we will not talk about the fundamental and applied aspects of ovarian cancer. We'll limit it to just about the metastasis of ovarian cancer. And what I'll do is I'll endeavor to introduce this disease to you. It's not a disease that, or it's not a cancer that experimental biological community in general is very familiar with. I will also try to flesh out some of the peculiarities of this particular cancer, which basically that itself teaches us that not all cancers are same, and therefore the mechanisms that drive the progression of one cancer need not always be relevant to another one. Before I begin, the idea or the sort of philosophical approach of my group is to look at these structures that form within our bodies. Now, these are breast cells. On the left hand side, it's fine. On the left, you have what is called an acinus, which is a bunch of non-cancerous breast cells that have come together within matrix. You might want to keep it off. The fluorescence signals go away. Or, oh, you want to take a picture first, isn't it? Whatever. Anyway, I'll keep going. And forms a structure which is very similar to what you get in the body. And if you were to replace these normal cells with cancer cells and put them within these same three-dimensional confines, they start doing what cancer cells do, which is you get a loss in that pattern that you get where you have these cells nicely arranged in a structure such that if you give them the right hormones, they will actually start secreting milk proteins into this hollow inside, just the way breast cells should be doing if those hormones are there in the body. And then you start losing this architecture and you move towards newer and newer sort of architectures as cancer cells start exploring their surroundings, start moving out, start dividing. And therefore, as a group, we try to look at this as a problem of pattern formation, where cancer cells typically tend to explore new rules of interactions with their surroundings and with themselves. And so cancer, just like development, can be looked upon as a problem of pattern formation and morphogenesis, where you are just having, as Darwin would quote, endless forms, most beautiful but also most odious in the way they of course affect us in terms of being a human condition, cancer that is. Cancer metastasis can be looked upon in sort of 4 spatial locales in general. One is of course where cancer cells they become transformed and then they move out of their primary focus of origin. And so experimental oncologists, as cancer biologists are called fancifully, obsess themselves over cells of origin, because an organ of course consists of multiple cells. So which is that one cell that becomes cancerous and gives rise to that particular cancer is a question that we frequently spend hours and years pondering over. The second part is where these cancer cells, once they come out, start entering into environments where they can get access to nutrition and move around all through the body. This is called intravasation, where they move into what are called endothelial or mesothelial compartments, environments. Endothelial are of course blood vessels, lymphatics. Once they seek these vessels, they move in there, they travel with the flow, and they reach other parts of the body. They have to migrate within these environments, and they are subject to different kinds of stresses, fluid stresses, shear stresses, stresses of being able to deal with immune cells, and so on. And finally, for reasons that are best known to themselves, and of course to hapless normal organs where they have eventually reached, they decide to settle down and start growing in these organs. And that process is called colonization. And these are the 4 sort of theatrical locales where several decisions are taken, several interactions are fraught, and that leads eventually to the homing of cancer cells from the primary site to the metastatic locale. A bit of show off with publications. What makes ovarian cancer very special or unique in some respects is the fact that when you look at these cells as they float around within those circulatory environments, they tend to be clusters more than single cells. Okay? And these clusters are called multicellular clusters by those who are not given to jargonry, and called spheroids by biologists. So these spheroids tend to, we've seen that these spheroids tend to be of different types, and at least 2 types, broad types, can be looked upon when one sees these spheroids under a microscope. So this is, for instance, the images, the non-cartoon images of these spheroids as they have been taken from the abdomen of a patient who suffers from ovarian cancer. Sorry, this is I'll just stand on one side, save you the trouble. So ovarian cancer starts from the ovaries, which are out here, and move into this circulatory compartment, which is called the body cavity or the coelom, okay, or peritoneum. Within this cavity, we saw, and so have others, that these cells tend to be not single cells, but clusters of cells, the spheroids. What we were able to identify further on is that these spheroids would not just be clusters, solid clusters of cells, cells that are essentially clumps of these cells, but also would form these very interesting structures which has a hollow within it. So row of cells with a hollow inside. Very similar to that acinus that I was trying to draw towards. But keep in mind, the acinus, which I showed in my first slide, was a non-cancerous organ-like architecture where you have cells which are responding to endocrine cues, responding to anatomical cues, and functioning physiologically to form a structure the way it would form in the body, like a breast or a mammary gland, and secrete milk and so on. These are cancer cells. They're supposed to have departed from that architecture, but they seem to have recapitulated an organ-like architecture within a fluid environment as they are floating around. This is a scanning electron microscopic image of the clump architecture, what we call, harking back to embryological terms, moruloids, morula-like. And some of you may remember from your developmental biology lectures, a morula is an embryonic structure where cells have achieved a 16 to 64 cell mulberry-like. That's what a morula is, mulberry-like appearance, like a bunch of grapes appearance, and that's what it is. How does the hollow spheroid look? It looks something like this. Of course, we are looking at the surface of this using the scanning electron, but you can appreciate how there seems to be some kind of a sheet or a covering that now seems to cover these cells, and you can no longer see those furrows which were so evident in the previous image. And this again is a very interesting feature, because for some of you who have taken cancer biology one-on-one, the basic idea of cancer again is to break through matrices. Matrix meaning the goo that surrounds cells. And they are supposed to secrete these matrix metalloproteinases, enzymes that cut through proteins, break through structures, allowing for the cancers to move. This seems to show that cancers not only destroy, they also can construct. They not only can break through matrix, they may also make matrix. Here is a spheroid that we imaged once the spheroid had settled down. After that had formed that hollow structure, it had settled down, and once it settles down, the spheroid starts disaggregating and then forming a sheet. So one can almost think of this entire process of metastasis as formation of spheroids within fluid compartments, traversal of these spheroids, and ultimately homing in them, adhesion, disaggregation, a new site, a new land. Typical colonization. They seem to break through the sheet or the shell as they come out, and you can almost see the cracks coming through, and you can see the furrows now visible as this shell breaks and the spheroid moves on. Another analogy is the implantation of the blastula, and hence we call them blastuloids, is the implantation of the blastula as it breaks through the uterine endometrium and then starts digging in towards, and there are scary parallels between implantation process and metastasis, which has been systematically looked at by a bunch of investigators very recently. So this is the process. And so, using classical cell biological techniques, we are able to identify what some of these proteins are, and lo and behold, they come to be the same kind of extracellular matrix proteins that are typical to those that cover normal, non-cancerous, gland-like epithelial cells called basement membranes. They form these shells around normal cells, giving them polarity, stopping them from moving, stopping them from dividing, allowing them to function physiologically, which in the case of the prostate would be to secrete the prostatic secretions, which in case of the sweat gland is to secrete sweat, which in case of the breast is to secrete milk. In this case, of course, they are secreting none of these, but the basement membrane even here is present. So in other words, cancer cells under stresses, under certain microenvironmental contexts, have recapitulated physiological, anatomical programs in order to tide over certain events, in order to perform certain functions, and for some purposes which we can go into subsequent. A bunch of molecular fanciful stuff, but we move on. looking at the integrity of these structures, what we did was we tried to see whether the blastuloids or the hollow structures are indeed much more integral, are morphogenetically more stable than the moruloids. And the way we would do this is we can take a moruloid and we can shower it with single cells and see if these single cells get integrated within these structures. It's easy to see integration, it's hard to see exit, but it's easy to see integration. And we could see that yes, if we were to take green spheroids, put red cells in them, they get integrated. That does not happen in case of the blastuloids. The blastuloids are able to resist entry and exit of cells. Very much, again, organ-like entities. If you take two moruloids, you put them together, they can coalesce. You can almost see this, where a green and a red moruloids have coalesced, and you can see a boundary. On the other hand, blastuloids remain separate from each other, even if brought close and allowed to date. Again, harking back to the idea of organs remaining separate, although they can be as close to each other as possible. A liver stays very close to a pancreas, or a gallbladder stays even closer to a liver, but they don't choose to mix with each other. There are organ-like aspects which keep organs separate, and we see that in the spheroids as well. What happens if you take this basement membrane or matrix out? So we can wash these. Okay, it's working. One more. Yeah. If you add collagenase, if you add an enzyme that can remove this sheet, they move back to a moruloid status. And if you wash that enzyme out, now you can see the kinetics of the hollowness coming back. Typically, biologists, when they see such hollow structures, especially in case of cancer, think of it as an outcome of necrosis, as an outcome of the fact that cells are no longer able to get oxygen, nutrition, and hence are dying. Doesn't seem to be the case here. This is happening over 3 to 4 hours. We can actually time the formation of these hollows, and it's very clear that the hollow is forming because of some other mechanism that allows cells to rearrange themselves back into this hollow state. My 15 minutes are over, so I'll try to wrap this up in the next 5 minutes. As cell biologists, we obsess with the hows, and how does this hollow form? What is the protein that is there at the edge of the cell? Where did that protein go? What interacted with that protein to give rise to it? What if I do a CRISPR and knock it out, or shRNA to knock it down? So I'll spare you the painful details of all of these, because the hows are fun and they take up a lot of time, but they're not as satisfactory as whys. Why is it that we have these two different types of spheroids? And some answers towards that came from one experiment where we asked, okay, so the question, so remember this other sort of concept that cancer biologists harp on and obsess over, which is called the intratumoral heterogeneity, phenotypic heterogeneity, right? Heterogeneity of much of that relates to cell states, epithelial mesenchymal, jammed, unjammed, and so on. Although jammed, unjammed is more of a multicellular phenomenon. The epithelial mesenchymal is a very single cell state spectrum, proliferative versus migrational. Remember, this is heterogeneity at the multicellular scale. You have multicellular clusters showing different phenotypes. The question is again, why? Moruloids, the clumped cells, the solid cells, tend to be more adhesive. They tend to settle down much faster than the hollow spheroids. They are able to form and immediately settle down. Fast colonizers. Okay. On the other hand, blastuloids have very interesting properties. Atomic force microscopy led us to look at their ability to be more elastic and less viscous in their rheological properties. And that allowed us to conduct these microfluidic experiments where we would mimic what is called stomata. Yes, I use the word stomata, which comes from botany, right? These are holes through which I think transpiration takes place in plants. So we too have stomata. In that abdominal cartoon, if you look at, we too have stomata through which fluid moves into the body cavity and moves out. And it's these stomatas in which, along with the fluid, cancer cells tend to get stuck. And that's how colonization occurs in those stomatal areas. Now, if you were to mimic these stomatal spaces, as we have done with the right kind of sizes, if we look at how these spheroids go through these stomatal-like confined volume spaces, we see that moruloids do very poorly. They tend to deform and they tend to break and disintegrate. Whereas the hollow, elastic blastoids can very easily remove the fluid out as they move through, come back and regain their aspect ratios without any seeming disintegration. We feel the lumen, and of course a bunch of other molecular details that one can add, imparts these rheological properties to these hollow spheroids, allowing them to be sensitive and resilient to more volumetric confines in order to explore body spaces to far greater extent than the moruloids. And so the why is that the moruloids are the slow colonizers, are the fast colonizers. The blastoids perhaps have the ability of colonizing over much longer time scales. So these are dual strategies that cancers use. Remember, patients have both these kind of structures to different extents. And so these dual strategies are used by cancers as means of using their phenotypic heterogeneity to be able to metastasize along different time scales. And that's the reason why, while chemotherapy can be given to a patient, there are resilient niches within the body which are able to resist these chemotherapeutic insults and tide over long periods of time and then ultimately metastasize. So I will end with this and and acknowledge the work of graduate students as well as undergraduates who worked hard at this project. Siddharth Botra, a senior of the undergraduates who are out here from IIC, is headed to the University of Chicago, where he was the one who built these microfluidic structures along with a graduate student and was able to observe these structures, especially for cells where we had done some molecular perturbations. And Monica Saravanan, who's currently doing a PhD also at the University of Wisconsin-Madison, again another undergraduate from our program, was instrumental in building computer models to basically decipher the fact that these structures need to have greater resilience in order to move through these volumetric confines. So with that, I'll stop and take any questions that you have before we break. Thank you.
So when you were comparing the moruloid and the blastoid while passing through a structure, right, you see that the blastoid breaks down into different structure, or is it that there are separate blastoids there?
No, there were two blastoids. They both came together. The blastoids, they don't break at all. They show very little breaking. All they do is they sort of seem to implode. They're able to squeeze out the fluid from within them, move past, and then regain their shape by again pulling in the fluid in. We are trying to look at what leads to this movement of fluids inside and outside, but they're able to do that under such conditions. The moruloids are the ones that almost get ground because of their movement, and we know, we've shown data where moruloids have cells, because again they are more morphogenetically labile. They can, the fact that they can accept cells and the cells exit out shows that the intercellular adhesion is very weak in moruloids, and we could show that using immunocytochemistry and a bunch of other things. And that allows these intercellular junctions to break far more easily in moruloids, allowing them to get deformed and even break.
So, sorry, one more question. We see in the initial phases of the blastoid movement, we see an acceleration. Is that because of physical pressure being applied, or is it because of adhesional forces?
Oh, this was, if you're talking about the video, that was just a pump running. So as of course it will come out, the flow is again maintained, so it will get sped up faster.
Under which cases are the moduloids get the advantage over the blastuloids?
Sorry?
Under which cases are the moduloids get the advantage?
So the moduloids get an advantage over the blastuloids when they are in a big, spatially unconfining space. So the moduloids are the ones which are actually attaching very fast onto surfaces that they see. And that's the reason actually why when we tap this fluid out from patients, and we do get access to this fluid and the spheroids from patients, much of those spheroids are blastuloids because the moduloids have already attached.
You said that there's a scary similarity between the implantation of embryos and implantation of cancer cells. Could you elaborate on what implications that might have or what we can draw from that?
Yes. The implications that I can draw from this is that there's very little that's romantic about implantation. It's a veritable battleground where actually the tissue of the mother gives it all and fights the ability of the fetus to get it implanted, and that is and the mechanisms that are used, which include enzymatic wars. You have trypsin, antitrypsin being loaded onto each other, are strategies which are very similar to the same strategies that cancer cells and normal cells that are battling it out employ, which basically comes again to the point that two tissues within our body have inherent properties of being able to resist each other from gaining ground, and that has again not much to do with any of the immune access that immunologists would sort of bring in as a way by which you can mount defenses against cancer and so on. So basically there are these sort of, how would I put it, natural defenses that all our tissues have to resist competition from other tissue players.
Thank you.
So has there also been a study of the internal processes in the cancer cell that might lead it to resemble the behavior of a developing embryonic cell? There might, I suppose there might be some pathway that is triggered that resembles the initial pathway followed in.
So there's been a lot of work. If you're interested, you might want to look up the work by Kshitij Gupta at Connecticut, University of Connecticut, and Gunther Wagner at Yale. They have done a lot of Vincent Lynch and Gunther Wagner. Vincent was, I think, a postdoc with Gunther when he did this work. So there's a lot of work that they have done out there where they have been looking at, now, okay, fine, metaphors and metaphors. What are the molecular links? And so there are molecular similarities that are also played out between these two processes. In fact, surprise of surprises, when we were looking at the glycoproteins that are typical to spheroids of ovarian cancer, we found proteins like zona pellucida 1, zona pellucida 2. These are the coats precisely that coat the blastula or embryonic blastula in mice and humans. So there are molecular parallels. Cellular parallels, of course, I showed you. There are strong cellular parallels. Molecular parallels also exist, which serve to similarize the morphogenesis in these two systems.
One question, sir. So we did see.
Not sir, Ramrat.
Okay, it's fine. So we did see that the moruloid cells and the blastoid cells are morphologically very different. But is there a chance of them changing morphology later?
Yes. So moruloids tend to transition into blastuloids eventually. It's a very interesting process that we see. Not all the moruloids do, and that I reckon is down to the heterogeneity of the cell states. So when three moruloids form, not all of them will have the exact proportion of the right kind of cells that you require. But in general, most of the moraloids, once they form, over time, if at least what we've seen in culture and in mice, if you keep them long enough, they eventually transform into blastoids. So the moraloids therefore have two fates. One, either once it's formed to settle down and start spreading, or if it waits long enough, it will become the blastoid and it will settle, it will stay in that cavity for a long, long time until its time comes to again do something. But here's the interesting thing. Work with our collaborators at NCBS actually shows that you can even have transitioning between moraloids and blastoids, and there are certain situations that we now know of where if we have the right kind of chemical context, even blastoids can transition into moraloids. And so the two states can